the lecture, which is a series of lecture. This is number 3, describing HIV immunopathogenesis, easy and fast. So these are the structure of HIV. In order to explain this, we have to understand the structure. We have the lipid membrane derived from the host cell that is basically anchored via glycoprotein 120 and 41. Then inside you will have the matrix protein, which is important, which is found in here. Then you have the nucleic capsid containing various enzymes, integrases, reverse transcriptase, and proteases, and single-stranded RNA, 2 copies. Now let's move to the life cycle. Life cycle is very easy. So first of all, you have the glycoprotein 120 will come into play here. Glycoprotein 120 will bind to CD4, and it will lead to conformation change that will allow it in the same time this glycoprotein will bind to CD4, then it will move, conformation changes, and it will also bind to the co-receptor early on infection, CR5, early on infection, okay? I mentioned it in video number 1. So this viral envelope protein binding to it, it will lead to conformation also changes. See here, binding, conformation changes, binding, conformation change, and lastly it will allow the glycoprotein 41 to come inside and make the fusion process. So the first attachment is mediated via 120, fusion is mediated via 41. Going inside, there will be uncoating of the nucleic capsid. There will be basically all of the content of the virus will be outside, including proteins and enzymes. The reverse transcriptase, which is found inside here, it will play a role in reversing the single-stranded RNA into double-stranded DNA. So that's why it's called as a retrovirus. Then you will go, there's a nucleus import, so that it will integrate into the nucleus, as you can see here. Now once this integration happened, so this is the double strand of the DNA of the virus. Once this happened, it will be infected for life. This cell is infected for life. Remember that this is CD4 or macrophage. Late on infection, early on infection will be macrophage. How did I differentiate it? Via the co-receptor. Okay, so there will be transcription, but also here, first of all, it will give us a genomic RNA, and then some of it will give me mRNA. mRNA will basically make these nucleic exports here. It will go outside, and you will have a mRNA and genomic RNA. Genomic RNA, which you can see it in here, as you can see here. Now, you should know that there is an enzyme known as proteases that is very important, because this proteases, now this virally protein, they are precursor protein. How they will form for us the enzyme and the various components? Via something known as a protease enzyme. Protease enzyme will basically, after translation, will work here, okay, in order to make it functional viral protein. Now, why it's called here maturation? That means this is immature. Why? Because the protease will work on the protein, even though they have been assembled, but these are precursor. They will work until they will form the whole structure here. And that's it. This is all about the maturation, and they will be putting a maturation. And this is all about the life cycle. Thank you.